All right, a lot of excitement in the room. I'm excited too. I'm excited to see kind of what y'all come up with this week. All right, so I'm going to talk for a bit just to get you kind of oriented and get you thinking in terms of how ambitious you want to be in the rest of the semester, and just kind of set your expectations and just give you some tips in terms of how to approach the remaining weeks so that things go as smoothly as possible. And they'll give you some time to make your teams, and they'll give you a bit more information about how to do the proposals, and you have the rest of today's lab and the rest of today's lecture, as well as the labs this week, to come up with your proposals, come up with the ideas of what you want to do for your research project. Okay, so I'm going to give you a bit of an overview. All right, so basically, right, the point of those first 4 weeks of doing the real-world problem projects was really just to give you what you need in order to do these research projects, right? So the stuff you've been doing so far in order to learn how to like look things up on the internet and on in the library, in order to kind of look at primary literature and review articles, in order to help narrow down your hypotheses and see what hasn't been done and how things have been done in the past, right? That's stuff we worked on these past 4 weeks that'll be very useful in the coming weeks. Also, you've done a lot of experimental design and gotten feedback on your experimental designs to find ways to make your controls more appropriate and how to design your experiments so that you know exactly what you're going to do when you get to the lab, that kind of stuff. Right? You've also learned kind of hands-on techniques, working with aseptic technique and finding ways not to contaminate your samples and your treatment solutions. You also learned how to analyze data in a basic way, and also learned kind of at least how to start writing some methods and results. Right? These are skills that can help you kind of when you're planning an experiment, know what results you're shooting for and what things you need to keep in control and balance between your treatments and controls. Right? So these are kind of hopefully skills that you're feeling much more confident with now than you were at the beginning of the semester, and that you'll get more confident as you use them in a broader way in these coming weeks. Right? So today basically I'm going to go through the general goals as well as the specific timeline for what we're going to be doing for the rest of the semester. And they'll give you time to make teams just to take a break from me talking, and they'll give you the specific guidelines for what you're doing in terms of proposals this week. Okay, so teams and proposals is the main goal for this week. All right, so general goal for your research projects and kind of for the whole course is, again, to have you experience sort of like a mini grad school experience, right? In that you'll be working, in a small research team in a group of 4. There will be one team of 3 per lab, but I want you to focus on making teams of 4 first, and then if there is a team of 3 that happens to be really passionate about one idea, that would be the team of 3. But for now, I'm going to start with teams of 4. But you won't be working on your own, right? Because you will be part of a larger research lab. From now on, every lecture, there will be time set aside so that those who want to present what they've been doing so far or ask a few questions saying, we're kind of stuck on this, any ideas, and just get a few minutes of feedback from the class. So we will be supporting each other as kind of a research lab would, right? So our class is going to be your lab mates, your lab family, essentially. Right? You also have access to the broader scientific community. I mean, our poster session, we are going to be sharing and getting feedback and ideas from the broader scientific community at the end of the semester. But you'll also be doing so, as you're planning your ideas this week, you'll be looking up what others have done online and drawing from others' scientific research and knowledge as well to help guide your research as well. And of course, you also have help from myself and Elizabeth as your supervisory committee to guide you along the way. Right, so to remind you of kind of the analogy from the beginning, right, the point of this course is to get you to actually take the tools and start using them, right, in a creative way. So we started off with something really simple with our fly experiments, but with each project, you've been giving more and more flexibility to try some different things, different mixtures, more complex experiments. And this research project is your chance to do something bigger, right, something that takes more than just a week or more than just one experiment to solve. Okay? So a few tips to keep in mind just to kind of get you, the things we were doing for the first 4 weeks were a little bit of a crash course, a little bit of, you have 30 minutes to think of something, go, right? But now that you have a bit more time and a bit more space and a bit more knowledge, you're going to be looking broader, right? So a few tips to how to kind of handle the freedom you're going to have in the next few weeks. One is to stay kind of scientifically curious. Don't get too focused on one idea and ignore everything else. Do pay attention to what's happening throughout your experiments. And if something kind of new and interesting comes up, feel free to change gears, right? So if you're doing one experiment and that experiment gives you some results, but then you notice another pattern in your results that seems way more interesting, feel free to change gears and study that instead. Right? That's kind of one of the benefits of kind of doing academic research, right? Because broadly, academic research is research that people would do in institutions like SFU, where the professors are free to research whatever is interesting to them. As long as they can find funding for it, they can research things that have no kind of direct financial benefit to it, right? Unlike other kind of private labs where they'd be specifically trying to design a kit for testing for COVID or design some sort of pesticide for killing some sort of insect, right? Academic research can go in whatever direction people are interested in, right? So that's kind of where we're at right now in that what you're doing, you're not trying to make money with it, you're just trying to learn something interesting and new, right? So as you're doing your first experiment, right, notice any observations or patterns that you can, especially in the early experiments where you still have 2 or 3 more weeks to try something different, right? And keep your mind open to changing direction, to exploring new things as they arise, okay? You also want to practice kind of the good habits that we've been sort of talking about, but you're going to have more time to actually use them, right? So do the kinds of things a grad student would do. One being planning experiments very carefully. So I mean, in the first week, you had like no time. You basically just threw some ideas together to try it, right? But with our second experiment and even the third one, you did a good job of kind of looking into the research to see what's been done before, so you're not repeating something others have done before. And you also were able to start looking at different methods as well. Some people started looking at, okay, so I want to study this particular essential oil. What concentration did other people use that's relevant? Right? What concentration is high enough to make a difference or low enough to not make a difference so I can aim appropriately? Right? So doing that kind of background research makes it more likely that when you actually do your experiment, that it will kind of tell you something interesting, right? Whereas if you just randomly guess at a concentration, you'd be less likely to get the results you want. Right? Another way to make sure the experiment is as likely to show you something interesting as possible is to have lots of discussions, right? Discuss a lot with each other, but also with myself and Elizabeth very, very often, right? So several times while you're planning, while you're doing the experiment, when you're done the experiment, do talk to us a lot. That way we can kind of help you catch mistakes or think of things in advance, so you're less likely to do an experiment and realize afterwards, oh, we screwed it up, we're gonna have to do it all over again. Okay? It's not the end of the world if you do, but the more we can catch things in advance, the better, right? Again, make lots of detailed observations, and as you noticing things, like let's say that when you're setting up, you notice something's happening with the seeds, or you took a peek at the seeds earlier on and something weird is happening, do talk to us along the way, right? Because the more you can kind of learn from one experiment before you start the next one, the better. Right, some of you might want to overlap one study versus the next because you do need to request materials in advance, so you might not always be able to do this, but generally speaking, if you're going to be going from one experiment to the next and letting the first experiment guide your next one, right, the more you talk about your first results, the better your next plan is going to be. Okay, any questions or concerns so far? Okay, other tips. So yeah, do talk to us, each other and us regularly. The reason why I'm going to set aside some lecture time for lab meetings every week is so that any group that's kind of in the middle of something or trying to figure something out or just have some new results can just say, look, this is my first graph for my first experiment. What do y'all think? We're thinking of doing this next. Any new ideas that we should try instead? Any changes we should do to the new experiment? Right, this is something that we did regularly in Gerhard Griess's lab, was we would have lab meetings at every major turning point in our research. If we're going to present something to a conference, we would definitely show it to our group, but also quite often we just have a lab meeting where anybody that's changing gears from one set of experience to the next will talk about what they've done, what they're planning next, and they get feedback, right? It's good practice for the person up front to get feedback and learn and benefit from everybody else. It's also good practice for everybody in the lab to be able to think about other people's projects. You're basically doing the problem solving of 12 different projects at once, and you'll learn a lot more than if you just worked on one thing by yourself. Right? So I'm going to encourage you to present to the class. When I say present, I don't mean PowerPoint. I mean like come up here in 2 minutes, tell us what's going on. We talk about it for a couple more minutes, sit back down when you've had enough. Right? So very, very casual. Right? But the more you can talk to people, the more you'll get out of your data, and the more you kind of can plan your methods in a more efficient and effective way. All right? Generally speaking, you want to correct mistakes before they're made if possible, but as quickly as possible. By asking questions and talking about it so that you don't end up kind of repeating an experiment that doesn't work. Okay? Another important tip, right? So you've been limited in terms of the number of replicates and tubes and plates you're allowed to use, right? Just partly because we're limited in space if all 12 groups are doing the same project, right? But you'll have more room to do larger numbers of tubes and plates and cages and stuff like that once you're like one of only 4 groups working on something. Right? So one thing I want you to keep track of is to figure out more efficient ways to do things, right? Because it really does multiply. For example, let's say that you're doing like the bacteria experiment and you find just some way to organize things or to arrange things with your team so you save about 30 seconds per tube, right? Well, in some experiments, you might be setting up like 120 tubes a day. Maybe not for this course because you only have 2 hours, right? But as a grad student, it's not unusual to set up 100, 200, 300 tubes a day for your experiments, right? So if you can save 30 seconds off of 120 tubes per day, you're saving an hour a day, right? And if you're saving an hour a day for your whole kind of graduate school career, that's like 2.5 months of saved time just from that 30-second savings, right? So if you can just find 4 or 5 ways to save 30 seconds at a time, right, you can cut like a whole year or 2 years off of your grad studies. All right, so look for ways to make things efficient. Once you have things maximally efficient, feel free to listen to your podcast as you do things and do things fast, right? But early on, which is for you, everything's gonna be early on, find faster ways to do things. Or if you're not sure, if you're like, this is taking forever, Kevin, how can I do things faster? I'll say, here's a bigger pipette. Let's just use that instead of pipetting 15 times, right? So talk to us if something seems to be taking too long, right? Tiny savings can save you hours, if not years, over your time, over your research career, all right? Also, things to keep track of. So now that you have a group that you'll be sticking with for the rest of the semester, right, we'll get you to stay in one area, and we'll also assign some drawers that you can use from now on to store whatever you need in between experiments. But then since there's other groups working in that same space in the next lab, you need to make sure your area is fully cleared, and any equipment that we share, you clean and put back before the end of each lab. Right? So please be considerate of your classmates and make sure everything is clean and stored properly before you leave the lab. Okay, any questions or concerns about any of this? Okay, all right, so let's talk about the timeline and some guidelines for how to do the research projects, okay? So all of this is in your worksheets, so I'm gonna kind of walk you through it here. So what I've been talking about is just like the first, let's just stay on camera, is like the first page, half page or so of these worksheets. If you look at the timeline, I'm just gonna highlight a few things for you to notice, right? So for most groups, you're gonna spend this week coming up with your general kind of, not rough ideas, but your outlined ideas for each proposed project. And then next week, you're gonna take the one that we choose, that we approve, and actually plan out the methods, right? So most of you are spending 2 weeks planning your experiment and then starting your research in week 7, all right? So the specifics of that is today, make teams, start coming up with rough ideas, and just run your rough ideas by us. By rough idea, I mean, hey, what if we try putting this and this in the cage with the flies? Like that's rough, and that's enough information for you to run it by me or Elizabeth to get some feedback. Okay, so super rough ideas, just 1-sentence idea, run it by us constantly throughout the rest of today's lecture. And then what you want to do is for each real-world project topic, come up with at least one idea. Right, so if you have a good idea for the fly project, you're like, check, okay, that's one that all of us would be happy to work with if it gets chosen. Let's keep that for now, move on to the plant project, right? And discuss and look up stuff online, take all the brainstorming y'all did for your self-reflections last week and just everybody read out their list and then pick a couple they're interested in and run it by us, for example, right? And once we say, oh, those 2 ideas seem really cool, maybe keep those for now, then move on to the bacteria project, right? And then look at all your brainstorm stuff, talk about which ones you like, and then pick a couple of things down and run it by us as well, right? As quickly as possible, you want to come up with 1 or 2 ideas per real-world problem topic, okay? Once you've done that, then you can start planning out the rough methods for each of those, right? Because the proposal you're handing in by the end of Thursday's lab, we'll talk about it, but essentially you need to have outlined kind of like a really rough flow chart of what you would actually do for your first experiment. So we know what kinds of materials and tools you're going to need. So we know whether we can have a certain number of projects of the same type without having everyone cramming into the same equipment in the same locations. Okay? So that's kind of the goal for this first week, is to run rough ideas by us first, to very quickly narrow down to a few ideas that you want to focus on, and then start designing the methods. And as you design the methods, you'll probably find that certain topics are too hard or too easy or not really realistic, and you'll chop some along the way. Okay? So each group will have 3 to 5 maybe ideas that you end up proposing, okay? And then next week you will actually kind of finalize your method. You'll make a request for materials as quickly as you can so that within week 7 or 8 you start doing the research, okay? So I'm trying to give, because working with teams is hard to find time where all of you are free to meet, I'm trying to give you as much class time as possible. So the rest of lecture, all of Tuesday and Thursday's lab is going to be figuring out ideas with your team, okay? As far as homework during this time, this week I just want you to focus on coming up with the ideas. That's a really important thing to do this week. Next week, while you're finalizing your methods and just waiting for the materials to be made, right, you'll have a bit more time in the evenings where you're not looking up stuff online. I want you to be doing your first self-assessments. right? You'll be looking back at the first 4 weeks of the semester and assessing your skills development, where you're at, where you started with, where you're at now, where you want to go by the end of the semester, that kind of thing. Okay? Any question about the next 2 weeks? Okay, so next page, page 2 here. So weeks 7, 8, 9, and 10 are your 4 data collection weeks, right? Some groups that might be super organized, maybe you'll start like in week 6, but most won't. Most of you probably need week 6 to figure things out and request materials, and you'll have your materials in week 7 or maybe 8 to start doing experiments. I'd say that what you want to aim to do is at least 2 experiments, right? You have 4 weeks, and you might think, oh, I'm going to do 4 experiments, or maybe squeeze in a 5th one somehow. I think realistically it's more like 2, right? Because you need to think about what you're going to do, request the materials, wait at least a week or so to get all the agar plates and stuff for Cheryl to make them for you, and then you'll do your experiment. And then next week you'll be looking at your results, thinking about what to do next, and request materials. By the time you have materials for the next experiment, right, we'll be close to the end of data collection, right? So 2 is realistic. If you plan ahead and request kind of more materials and have plans for each experiment, maybe you can squeeze in a 3rd or a 4th. Right? But we're looking at around 2 well-planned experiments to get as much information as you can. Okay? So you will kind of want to plan your experiments such that whether the results are what you expect or not, that you'll learn something interesting either way. That you have something interesting to present either way. Okay? So during the data collection weeks, a few things to notice. So the lab times are basically just data collection. You're either planning or doing experiments. Okay? Lecture time-wise, you'll notice that the first half of each lecture is going to be teaching you kind of more details on how to write each part of your poster, right? So week 7 is going to be methods and results, a bit more tips for that. Week 8 is going to be discussion and intro. Week 9 is a holiday. Week 10 is going to be all the remaining parts, abstract, title, acknowledgements, references, all that kind of stuff, right? So after I teach you how to do each of these sections, you'll basically spend the week with your team for homework, writing it up, right? So team write-up of methods and results. Team write-up of introduction and discussion, okay? As I said, the second half of the lab, the lectures is going to be lab meeting for whoever wants to present what they've been going, or just talk about what they've been doing, get feedback. That's open for that. You also notice that there's gray highlighted sections here. That's just an optional assignment if you're interested in peer reviewing other people's work, right? So the deadline for getting your intro methods, results, and discussion written is by the end of week 9. So if you're interested, you can I can assign you 1 or 2 other teams' intro methods, results, and discussion for you to give some written feedback on it. It's really helpful to get your target audience, which is each other, to look at your writing and tell you, oh, this is really clear, that's really good. I'm confused by this, what's going on there? Question marks, right? So it's really good learning experience to be both the reviewer and the person being reviewed. All right, so if you're interested, it's optional because it's not completely essential to your learning, but I think it's a worthwhile exercise if you want to do something extra. Okay, so after those 4 weeks of data collection, week 11 and 12, you'll be basically preparing your posters. So after week 10, that's it, no more data collection, right? So plan accordingly. Make sure you're starting things early enough that by the end of week 10, everything is wrapped up and cleaned up. All right? Week 11, you'll basically take, well, you already drafted most of the poster. You'll basically be taking all the feedback you got from Elizabeth and each other to make your kind of second last draft of your poster, right? So that before the end of week 11, you'll show us your draft and kind of do a practice presentation and Q&A session, right? Just to practice what you'll need to do during the poster session. And then once you've gotten some feedback on your second to last draft, week 12, you make one last round of improvements, print it out, lay it out nicely on a piece of kind of black poster board, and you're good to go, right? By week 13, Monday, we're ready to present. Okay, questions? Okay, so enough talking for me for now. So now, first big task for today, find your team of 4. Okay, so to keep things organized, if we can get like lab 1 people to stay on, is it usually this side? Yeah, stay on this side of the room, lab 2 people on this side of the room. Find your teams of 4, form the teams however you like, right? Generally speaking, I think choosing people you enjoy working with is good, right? You can find people who might have different complementary skills. Some people like hands-on, some people are good at writing, some people like researching things. If you have people with different skills and preferences, that can make for a good team. Also maybe find a team that's similar in terms of ambition, right? If you're basically really busy with other courses and you want to do something kind of straightforward that you know is going to work well, right? Maybe find teams with a similar kind of ambition. Or if you want to go really kind of ambitious and do something really crazy, then maybe find other people who are as crazy as you. right? However you like to do so, find your teams of 4. If you have your team of 4, just sit together and start coming up with ideas, right? I would say start by sharing your brainstorming ideas from last week, and then start narrowing down which 2 or 3 you want to explore a little further for each real-world problem topic. Once everybody has a team, then I'll call you back together to give you more advice in terms of how to do the proposals. Okay, but go ahead and form teams. Any questions, let me know. So it seems like everybody's found the group, so thank you for the people who had to be flexible to find the groups. Thank you. Okay, so I'm going to give a few more guidelines in terms of how to approach your research problem, your research project proposals, and then I'll give you set you free again just to continue running ideas by us. Right? Some teams have already started running the rough ideas by me and Elizabeth, which is awesome. Okay, so basic rule is, as I said, each team by the end of your Thursday lab needs to hand in at least one proposal per real-world problem topic. Right? Part of the reason is because we're going to want to spread out such that there's maybe about 4 teams per topic, so that we don't have 11 out of 12 projects be the same type of stuff, because that'll make for the proposal session be pretty boring. It also means that you'd run out of materials, because if we're splitting, say, the 100 cages we have across the 11 groups, you're not going to get enough replicates per week, right? Not enough space, not enough materials to get everything done. All right, so we want you to propose something per topic. Also, I want you to have a range of different ambitiousness, right? At least one of your topics should be something super easy that you're for sure going to learn something from and it's going to work, right? Because the problem we've had in the past is that people get super ambitious with their ideas, and if all 3 of them are very ambitious and very unlikely to work, we might not approve any of them, and then now you're a week and a half behind, right? So I want you to have at least one project that you're sure will be very easy to get done, and no matter what happens, you'll have a interesting result from, right? So try to have something ambitious, and then from there you can try to have something that you're pretty sure will work, and then you can have another idea that's a little more ambitious, another idea that's really wild, right? And try to justify it either way. All right. So in terms of what you're going to do for each proposal, this doesn't have to be something formal at all. This can be completely like I brought kind of crappy scrap paper just to send you the signal that it's just scribbles on a piece of paper, right? Is what I'm looking for. It doesn't have to be well written at all. Right, the main thing I want you to share with us is your idea, like your research question or your hypothesis that you're going to be exploring. Give us some reasons why you want to explore it, right? Even if it's just that some crazy lady on Facebook talked about something that you want to explore, that's fine as well. As long as you have a chance of learning something, whether it works the way you think or not, right? So, but basically just, your idea and why, which you've already probably run by us already to get some feedback. A flowchart. Right, so I want you to think through every step of your experiment and kind of draw it out in detail, or just write it out in detail, right? So this is going to be more detailed than your poster version ones, right? The feedback that Elizabeth and I have been giving in terms of, you don't have to tell us that you're going to mix this much water with this much whatever. I want you to go back to that level of detail for this, because the point of this flowchart is not to have a concise picture for your poster. The point of this flowchart is to make sure that you've imagined every step you're going to do and thought about, I'm going to need a beaker because I can't get the solution out of the small tube with a syringe. Right, so I'm going to need a beaker that's about 50 mils, and then I'm going to have to need a syringe filter in case it gets clogged. I'll need some extras, and then I'm going to have to take that solution. I'm going to have to put it into something sterile. I want you to think through every step that you're going to do and write it out. If you don't want to draw it, you don't have to draw it. Just write out step by step. Step 2, sterile filter in 0.45. Step 3, sterile filter in 0.22. Step 4, put this in whatever. I want you to walk us through step by step so that you've thought through every step. Right? Also tell us the number of replicates, right? So if you're going to do that for one treatment, but you're planning on doing 2 different treatments in your first experiment, tell us your 2 treatments. Tell us how many replicates you're doing. Because what Cheryl and I need, and Elizabeth and I need when we talk about your projects on Friday, is to get a feel for how much space and materials and equipment is your first experiment going to use, and also in your follow-up experiment as well, so that we can see which projects we can fit together into the lab. Okay? So that's what you need to have in your thing. It doesn't have to be clean, right? It can be just stick figures and scribbles, right? But I need to be able to see that you've thought through all the steps, you've listed the materials and equipment you're going to need, times however many replicates you plan on doing for your first experiment. So this flowchart is for your first experiment only, because presumably once you get the results from that first experiment, you might change your methods, or you might test something else based on what you learned from the first experiment. Okay, does that make sense? That's your proposal. Idea, a little bit of rationale, and then detailed steps or flowchart to tell us exactly what you plan on doing, listing your materials, and then times however many replicates, so we have a feel for how big your project's going to be, how big your first experiment's going to be. Okay? So to help you with that, a few different things. So let me see. Flowchart proposals. Okay. So what I have here, and I posted this on Canvas already, is my old equipment request form, which I updated this morning, right? So this lists all the equipment that I could think that you could possibly need, right? From like calipers to centrifuges to falcon tubes of different sizes, graduated cylinders, every size of pipette, the flies, the seeds, the essential oils, right? Everything I could think of is here. So when you're thinking, I also listed it such that I kind of wrote down whether I think you might need it for each particular real-world problem project, right? So if you're kind of thinking through the methods for your fruit fly experiment, what you can do is highlight all these rows and sort by row column B, and it'll list all the things that I think you need for this type of project, right? Those would be yes. The yes question mark, the things that you might need, like a cutting board, right? So you can go through that list and see, did I include these kinds of materials in my flowchart, right? Just to remind you of the stuff you might need while you're doing things, right? Because you might have forgot, oh yeah, if I'm going to cut up a dragon fruit, I'm going to need a cutting board, a knife, I'm going to need something to put it in, I need a blender to blend it up, I need cheesecloth to get rid of the solids, right? So this is just to inspire you and remind you of the things to think about, okay? So I have printouts of these, which you don't need to write on, right? Your actual equipment request you're going to do next week when you actually have a proposal approved. You're also going to do them electronically, so when you give them to me, I can cut and paste the rows and add up all the materials. I can know, okay, we need like 300 test tubes or something like that, okay? But I have a printout, I have enough for one per group that you can go through the list, right? So yeah, Elizabeth, maybe if you wouldn't mind giving out that. Okay, so in terms of how I'm gonna, so basically by by the end of Thursday's lab, you need to hand in your proposal. So you'll be handing at least 3 pieces of paper, one for each of your real world problem kind of projects ideas. But if you have like 2 ideas you couldn't decide between for the fly project, then hand in 2 ideas. If there's 3 ideas that different teammates are really interested in for the bacteria one, hand in 3. Right? There's no real limit to it other than the time and energy you want to put into it. So what we're going to do is on Friday, I'll sit together with Cheryl, with Allison, who's kind of Cheryl's understudy for this semester, as well as Elizabeth, and I will sit together on Friday afternoon at 2:30, and we're going to look through all the projects together. Right? So for your project, so let's say you have like 2 ideas for project 1, and then 1 for project 2, 1 for project 3. I want you to number them based on your favorites. Right? So the one you're most interested in might be a project 3 one, and then second choice is this one, third choice, fourth choice. Right? So that's going to be the first thing I look at is what's your first choice. Okay? Second thing I'm going to look at is, well, I'll look at your first choice and see, is it realistic? Is it likely to give you something interesting, whether it goes as planned or not? Right? If it's really unrealistic or super unlikely to work or way too complicated or unrealistic, then I might have to veto it and look at your next choice. Okay, so you want to have thought out your idea to make sure that it's something that's doable, and maybe just run it by us before you propose it, just to save you time for proposing something that is worthwhile. And then what we're going to do is take a look at how many people propose projects for each set of tools, right? Because if 11 out of 12 teams decide that they want to work with bacteria, right, we're going to try to pick the best 4 then, right? Pick the 4 that we think are most likely to work, the 4 that are most different from each other, so we don't have 2 projects that are almost the same. Right? The ones that are well planned enough that we think these people know what they're doing, so even though it's ambitious, they can get it done, right? And the more you tell us, the more clear you make it, the easier it is for us to make that decision, right? So that's kind of the 3 priorities we're going to try to balance is what you're most interested in, what we have space and the materials for, as well as what's different from each other's projects and what's likely to work, all right? So because we're going to be spreading people across the different projects, right, you could get your last choice, right? So don't propose something unless your team's actually interested in it, right? If you don't have something you're interested in for a particular real-world problem topic, look harder, okay? Or talk to us about, if you're completely drawing a blank, just talk to me or Elizabeth and we'll try to generate ideas together. But you want to make sure that for whatever 3 or 4 projects you produce, even if you get your last choice, you'll be like, that's cool, let's do it. Sound good? Okay. All right. So I think that's mostly what I have to say. There's more specific tips and stuff to watch out for on the actual worksheets. Feel free to look through those. But for the rest of today, I'd say run your rough ideas by us first to get feedback on those. And don't go into the detailed methods until you have rough ideas for all 3 real-world problem topics. Okay, so I think that's a better way to go. Because once you have that, then you'll be able to narrow down your methods more efficiently. Okay, questions? All right, go for it. So again, your ideas can be super rough, like a 20-second pitch. If you have that much, run it by us right away. What Elizabeth and I might do is kind of go from group to group to talk about whatever ideas you have so we don't miss anybody this time. Yeah, so maybe you start on that side, I'm going to start on this side, and we'll just take group by group, no matter what they have, whatever they have so far, talk about it and move on.